Hey guys, welcome you all back to this channel. And here we are now looking forward for NTSE preparation series, and we are in the second part of this chapter, that is cell, fundamental unit of life. Okay, I hope you all might have watched the first part. If you haven't watched that, okay, the description is there. Sorry, the link is there in the description. Just go through that and come back to this video. Okay, if you haven't subscribed this channel at now, okay, so please do the subscription, okay, and please share this video so it will be helpful for the students who are preparing for NTSE examination, Olympiad examination, as well as CBSE board examination. Okay, so fine, without wasting time, we'll start the chapter. And today we are going to study about all cell organelles. Okay, we are going to study about cell organelles. In the previous video, we studied about the introduction of cell, and as well as how the cell wall is made up of, and what will be the difference between cell wall and cell membrane. Okay, today we'll be start talking about a core cell organelles. Okay, so cell organelles are also called as cell members. Okay, cell organelles are also called as cell members. Okay, so when we talk about a basic revision about the previous chapter, you can say cell is divided into two types based on the numbers, prokaryote or eukaryote, or based on the presence. Okay, based on the presence where it is present. Okay, so if it is present in animal, then it is animal cell. If it is present in plant, then it is called as plant cell. Okay, so when we talk about prokaryote and eukaryote, based on the nuclear membrane only, we are going to differ whether it is prokaryote or eukaryote. Okay, so we'll start studying what is nucleus first of all. Okay, so nucleus is one of the important component. It is one of the important component of the cell which control all functional activities. That means it is going to act as a brain of a cell. It is going to act as a brain of a cell. Okay, so when it is acting as a brain of a cell, this nucleus is going to give command. Okay, this nucleus is going to give command for all other cell organelles. So for you can say it's kind of leader. Okay, so the leader is going to give command for all other cell organelles. So you have to do this work, you have to do this work, like that. Okay, so this was first discovered by, this nucleus was first discovered by Robert Brown in 1831. Okay, it was first discovered by Robert Brown in 1831. In he discovered a dense spherical body cell. Okay, he founded a dense spherical body cell on an orchid. Okay, he found it on an orchid. Okay, and named it as a nucleus. Okay, when we talk about the ultrastructure, it will be spherical or oval in shape. Okay, so it will be spherical or oval in shape, and most of the nucleus will be present in the center. So if you talk about animal cell, usually the nucleus will be present in the center, whereas when you are talking about a plant cell, the nucleus will be present in the periphery. Okay, so that is how it is going to be located. And nucleus is double membrane. Okay, nucleus will be double membrane. Okay, so you can say it will be covered with an extra membrane. Okay, that will be covered with an extra membrane. Okay, and that membrane is called as nuclear membrane, and that membrane is called as nuclear membrane. Okay, when we talk about the chief components of this nucleus, that means members of nucleus. Okay, so chromatin material is one of the member, and another one is nucleolus. Okay, then you can ask me what is chromatin. Chromatin is nothing but it is a form of intertwined mass. Okay, so chromatin material is nothing but your gene. Make it very simple. Chromatin material is nothing but gene. Okay, gene, or you can say DNA. Okay, so that will be coiled, and it will be looks in a thread-like structure. Okay, it looks in a thread-like structure. Okay, so this chromatin material mainly consists of DNA. Am I right? Okay, so this DNA is responsible for hereditary information. Okay, it is responsible for giving hereditary information. What is hereditary? Hereditary means transfer from one generation to another generation. Transfer from one generation to another generation is called as heredity. Let's say like your father may be having a character. The same character will resemble in you also. Okay, that means your father have inherited that character to you. Okay, so that character is transferred from your father to you. Okay, that is called as one generation to another generation. Okay, that character will be transferred by means of DNA. Okay, in the form of DNA. Okay, so this DNA, when we talk about DNA, let let's talk about DNA clearly. Okay, DNA complete full form of DNA is deoxyribonucleic acid. Okay, so DNA is full form is deoxyribonucleic acid. Okay, so this DNA is made up of a sugar. Okay, this DNA is made up of a pentose sugar. Okay, and as well as it has a nitrogenous basis. Okay, it has a nitrogenous basis and it has one phosphate group. Okay, so just have a idea about it. Okay, no need to go deeper about this. Okay, what is DNA? DNA is the genetic material. Okay, so this DNA will be present inside nucleus in the form of chromatin, in the form of thread-like structure. Okay, so they will be having, this DNA is made up of a ribose sugar. This is made up of a ribose sugar plus nitrogenous basis plus phosphate group. Okay, that is how DNA is arranged. Okay, so this DNA, when you see, it is helical in shape. Okay, this is helical in shape. You could have seen this. Okay, so this shape is called as helical shape. Okay, and this was given by Watson and Crick. Okay, the structure was proposed by Watson and Crick. Okay, so usually in a person usually 23 pair of chromosomes will be present. Okay, totally 23 pairs of chromosomes will be present. Okay, so chromosomes means two chromatin join together and form one chromosome. Two chromatin, that means let's say one thread, okay, and another one thread. Okay, two threads join together and form one chromosome. Okay, so when you say 23 pairs, that means totally 46 chromatins. 46 chromatins are present in a human. Okay, so these numbers are differed in organisms. Okay, these numbers, the number of chromatins or the pair of chromosomes are differed from each and every animals, organisms, like that. Fine, so this is all what about nucleus. When we talk about when we see the diagram, okay, so this nucleus, you can see this is a structure. Okay, the nucleus is covered with nuclear pore, which is a discontinuous pore. Okay, so they will be having a double membrane which has pores in between, so this pores is used for releasing genetic material. Okay, this is used for releasing genetic release. Okay, this is used for releasing genetic material from nucleus to the cytoplasm. Okay, so you can ask me why it has to be done. That is nothing but DNA, usually it has to be converted into protein. Okay, so your DNA has to be converted into protein for cellular process. Okay, for cellular process, DNA has to be converted into protein. So okay, fine, so that DNA has to come out through the nucleus, through the genetic pores. Am I right? Okay, and this nucleus is completely filled with a matrix-like structure. Okay, the nucleus is filled with a matrix-like structure, and that matrix is nothing but called as nucleoplasm. Okay, and this nucleus also covered with endoplasmic reticulum, and that have a nucleolus. Nucleus contains nucleolus, where exactly the chromatin materials will be present. Okay, and the nuclear envelope, it is of two members. Okay, it is of two layers. Am I right? Okay, one is inner membrane and another one is outer membrane. Okay, fine. So, and outside the nucleus, you can see your normal cytoplasm will be present. So this is what all you have to know about nucleus. Okay, so when we talk about the functions of nucleus, so nucleus is going to be the brain of the cell. Am I right? Okay, so which is going to control all metabolic activities. Okay, it is going to control all metabolic activities. Okay, so first one. Okay, next one, it also regulates cell cycle. What is the cell cycle? Cell cycle is nothing but life cycle, lifespan of a cell. Okay, so once the lifespan is about to over, one cell will be divided into two cells. Okay, so that means it is going to produce a daughter cells. Okay, so the parent cell will be producing daughter cells, right? Okay, so that process will also be controlled by nucleus. Okay, and it also be concerned with transmission of hereditary traits. Okay, it also concerned with hereditary traits. What is hereditary? Which is transferred from, the character which is transferred from one generation to another, that is from father to daughter, okay, or mother to son, okay, whatever it is, right? Okay, so hereditary transmission is also being controlled by nucleus. Fine. So this is all the functions of nucleus. I hope you all understood. I'm just giving a basic idea. What is nucleus? Nucleus is the important component of the cell which controls all functional activities. It was discovered by Robert Brown in 1831, right? Okay, it is spherical or oval shape. Okay, it has double membrane, and those membranes are called as nuclear membrane. Those membranes are of two types. One is inner membrane and outer membrane. Both are there. Am I right? So when you talk about the components, members of nucleus, chromatin material and nucleolus both are present. Okay, so chromatin material is the place where the DNA will be present. Okay, so this chromatin materials usually it will be coiled in a form of chromosome. It will be coiled in a form of chromosome. So totally 23 pairs of chromosomes are there in a human. That's it. This is what you have to remember for your nucleus. Okay, so PPT and all the notes will be in the description. Don't worry, you can take it later. Fine. So we'll move to the next cell organelle, which is another one important cell organelle called as endoplasmic reticulum. So next cell organelle is called as endoplasmic reticulum. In cytoplasm, some open and closed cavities will be there. Okay, you can say inside a cell, some open and closed cavity, a thread-like structure or whatever it is. Okay, so something will be present, and those cavities are bounded with some membrane. Okay, those cavities are covered with a membrane, and that membrane, that cavities are going to form a network. Network means chain. Okay, it is going to form a network, and that network is called as endoplasmic reticulum. Remember this is very important. Okay, so what is endoplasmic reticulum? In cytoplasm, some cavities will be present, which is double membrane, and they will be joined together and form a network, and that network is called as endoplasmic reticulum. Am I right? Okay, so who founded this? K.R. Porter identified this. Okay, he first reported in 1948. Okay, he first reported in 1948. Okay, so and he said it is a net-like system. It looks like net-like system. Okay, so joined together, right? Okay, that is how he first identified. When we talk about the structure of endoplasmic reticulum, so this endoplasmic reticulum usually it is divided into two types. Okay, this endoplasmic reticulum is divided into two types. One is rough endoplasmic reticulum and another one is smooth endoplasmic reticulum. Okay, so how can you say this is rough and smooth? With the help of presence of ribosomes. With the presence of ribosomes. Okay, here you can see this. This is called as ribosomes, which is attached to the endoplasmic reticulum. So when an endoplasmic reticulum is attached with ribosomes, those endoplasmic reticulums are called as rough endoplasmic reticulum. It is also represented with a term RER. Okay, so here you can see the smooth endoplasmic reticulum, which does not have any ribosomes. Okay, the surface will be smooth because of absence of ribosome. Okay, and those are all called as smooth endoplasmic reticulum. The surface will be very rough because of presence of ribosome. So it is called as rough endoplasmic reticulum. Hope you all understood, right? Okay, so rough endoplasmic reticulum will possess rough wall with attached ribosomes. They are rough in structure because of ribosomes. So what is the main role of RER? What is the main role of rough endoplasmic reticulum? Which involves in protein synthesis. What is the meaning of protein synthesis? To produce protein. Okay, synthesis means produce. Okay, so which helps in synthesizing proteins. And the next one, smooth endoplasmic reticulum, which involved in lipoprotein. Okay, so it will be helps in producing lipids. It helps in producing lipids and proteins. Okay, so this lipoprotein, lipid and protein together called as lipoprotein. Okay, and also it helps in synthesizing glycagon. Okay, it also helps in synthesizing glycagon. Okay, so this smooth endoplasmic reticulum plays a critical role in membrane synthesis. Okay, you can see a cell is there. Okay, the cell will be covered with a cell membrane. Am I right? The cell will be covered with cell membrane, and this cell membranes are made up of lipids and proteins. Remember this is one important question repeatedly being asked. Okay, so which material helps to produce or to like cell membranes are made up of which materials, okay, which substances? It is made up of lipids and proteins. Okay, so where the cell is getting lipids, the lipids are synthesized by smooth endoplasmic reticulum, SCR. I'm right, it is all produced from smooth endoplasmic reticulum. Okay, and this also helps in detoxification. That means when any toxin substance, when any harmful substance enter inside your body, any toxic chemical, okay, so which is harmful chemical, when it enters inside your body, that chemicals will be detoxified, okay, with the help of SCR. Okay, that is also the role of SCR. Am I right? Okay, so what is this? Let's go with the functions. It helps in supporting skeleton framework. What is skeleton framework? Membrane. Okay, it helps in membrane synthesis. Am I right? Okay, so it helps in membrane synthesis, and that membrane gives a skeleton framework for the cell. Am I right? Okay, and also there is some few enzymes which are present in endoplasmic reticulum, which will helps in synthesizing fat, steroids, and cholesterol. Okay, just know this, just look over this sentence. Okay, no need to go very deeper about it. Okay, for your level it is not that required. Fine. Okay, so enzymes will be present on the endoplasmic reticulum, which will be helps to produce fat, steroids, and cholesterol. So this is all about endoplasmic reticulum. Okay, so we'll have a small recap of endoplasmic reticulum. It is nothing but some cavities, open and closed cavities with a membrane. So this is all single membrane. Am I right? Okay, with a single membrane which form a network, okay, which form a chain, and that is called as endoplasmic reticulum. So it was first identified by K.R. Porter in 1948. Okay, and he found it in net-like structure. Am I right? He found it in net-like structure. When we talk about ultrastructure, two types of endoplasmic reticulums are there. One is RER and another one is SCR. RER will be rough because of ribosomes. SCR will be smooth because of absence of ribosome. Okay, RER involves in protein synthesis, whereas SCR involves in lipoprotein and glycagon synthesis. That's it. This is what we have to know about endoplasmic reticulum. Hope this is helpful. Fine. So if you are enjoying my lecture, okay, please do subscribe and like this video. Okay, that will be a motivation for me. Okay, and also share this to your friends, so your friends will also get to know about one of the better teacher who is teaching something good for students, right? Okay, we'll go to the next cell organelle, that is ribosome. See, so many members are there in a cell. Okay, so we are now completed cell wall, cell membrane, nucleus, and endoplasmic reticulum. Next we are going to talk about ribosome. Okay, so why? Because already we said this ribosome will be attached to our endoplasmic reticulum. Am I right? Okay, and also helps in protein synthesis. But this ribosome will not only be present in endoplasmic reticulum, it also present in chloroplast, it also present inside nucleus, and it also present inside mitochondria. Okay, so this is all the different cell organelles. We'll study all these things. Okay, so first we'll see what is ribosomes. Okay, so this ribosomes usually found in both prokaryote and eukaryote. Remember, this is very important point. Which organelle can be visible or which is found in both prokaryote and eukaryote? It is nothing but nucleus, nucleolus, or ribosomes. Am I right? Okay, and they are free form. Okay, they are in free form, that is called as monosomes. Remember, this ribosomes which will be floating. Okay, two types of ribosomes are there. One is free floating ribosome is there. Okay, free form ribosomes are there, and another one is attached ribosomes are there. Am I right? Attached ribosomes are there, as well as free form ribosomes are there. Okay, freely floating ribosomes are there, and those type of ribosomes are called as monosomes. Okay, all these monosomes will join together. Okay, will join together and will form a palette. Okay, will form a group of structure, and that group is called as polysomes. Group of ribosomes are called as group of ribosomes will form polysomes. Poly means multiple. Okay, group of ribosomes form polysome. Okay, so you can say this ribosomes are the smallest organelle of a cell. Okay, and this was first founded by first founded by Robinson and Brown, but they identified this ribosome in a plant cell. Okay, because Robinson and Brown were botanists. Okay, they were researching on plants. Okay, so they first identified ribosomes, Robinson and Brown. Whereas in animal cell, a scientist name is Palade. Okay, he identified ribosomes first. Okay, he found very small particles in a cell. Okay, and in an animal cell, and he termed those ribosomes named as Palade particles. Okay, he termed those ribosomes into Palade particles. Hope you all understood. Okay, so when you talk about the structure of ribosome, usually it will be very dense. Okay, I said now it is all going to be associated, joined together. Okay, or it will be spherical in shape, or it may be granular. Okay, so the ribosomes which is present in endoplasmic reticulum, those ribosomes are granular shape. Granules means like a stone-like structure. Okay, which will be attached over the endoplasmic reticulum. Okay, this spherical and all like free floating ribosomes. Okay, so this ribosomes also known as RNP particles. Okay, so it is also known as RNP particles. Ribosomes, whichever is dense, spherical and granular ribosomes, is also known as RNP particles. What is the meaning of RNP? RNP is nothing but ribonucleoprotein particles. Okay, remember this. It is also called as ribonucleoprotein particles. Ribosomes are also known as ribonucleoprotein particles, RNP particles. Okay, so they may be freely present or they may be attached. Am I right? Okay, so they are freely present inside nucleus, they are freely present inside chloroplast, and they are freely present in mitochondria. You can see, here in chloroplast, nucleus and mitochondria, endoplasmic reticulum will be free, whereas in ribosomes will be free, whereas in endoplasmic reticulum, ribosomes are attached. Okay, so this ribosome usually it is of two subunits. Now we are talking about structure. Okay, okay, let me erase all these things first. Yeah, so two units are there, two subunits are there. One is small. Okay, let's say this one is smaller subunit and this one is larger subunit. Okay, so always this units will be attached only. Always this ribosome units will be attached only, and that will be attached with magnesium ions. So all these multiple ribosomes will all attach together, and it will be called as polysomes. It is all called as polysomes. When you are asking, so tell me the difference between a ribosome which is present in prokaryote and ribosome which is present in eukaryote. Okay, so in prokaryote, the ribosome which is present in prokaryote is called as 70S ribosome. Okay, whereas in eukaryote it is called as 80S ribosome. Remember, prokaryote 70S, okay, and eukaryote 80S. Okay, so this 70S, when you are taking in concept of this larger subunit and smaller subunit, larger subunit have 50S and smaller subunit have 30S. Okay, whereas in eukaryote, larger subunit have 60S and smaller subunit have 40S. Okay, yes means Swedberg unit. Okay, yes means Swedberg unit. Okay, so this is the unit which is used for measuring sedimentation. Okay, which is used for measuring sedimentation. Okay, so that is called as Swedberg unit. Okay, no need to go deeper about that. Okay, just now in this class, you just remember what is yes. Okay, yes means Swedberg unit, which is used for measuring sedimentation, level of sedimentation. Okay, and two types of ribosomes are there: 70S ribosome, which is present in prokaryote, and 80S ribosome, which is present in eukaryote. Okay, 70S will be divided into 50S large and 30S small, whereas 80S will be divided into 60S large and 40S small. Hope you all understood what I'm trying to say. Any doubts in this? When we talk about the functions of ribosome, coming back to the functions. Okay, so this helps in, this plays an important role in protein synthesis. Already we studied, right? Okay, this involves in protein synthesis, and ribosomes are also called as protein factory. Okay, so they are also called as protein factories. Okay, where the protein are getting synthesized. Okay, that is why it is called as protein factory of the cell. Fine. So this is all what about ribosome. I'll just give a small recap of this. Ribosomes will be present in both prokaryote and eukaryote. If it is present in prokaryote, then it is 70 years. If it is present in eukaryote, then it is 80 years. Okay, and they may be free, they may be freely present, or they may be attached. Okay, they are freely present in chloroplast, nuclei, and mitochondria, where it is attached in endoplasmic reticulum. Okay, they are the smallest cell. Okay, the free form ribosomes are also called as monosomes. It was first identified by Robinson and Brown in plant cell, whereas it was identified by Pellet in animal cell. And Pellet termed those cells, those ribosomes, as Pellet particles. Okay, and these ribosomes will be also known as RNP particle. What is RNP? Ribonucleoprotein particles. It is also called as ribonucleoprotein particles. Fine. Okay, and they will be attached. Always they will be attached with two subunits. One subunit is called as smaller, and another subunit is called as larger. Okay, both subunits will be joined with the help of magnesium ions. Okay, ribosomes, multiple ribosomes will join together and form polysomes. This is all what you have to remember about ribosomes. Fine. So shall we move to the next? Yes. Next one, this is very familiar cell organelle among the students, because all might have studied about this powerhouse of cell. Okay, so mitochondria is called as powerhouse of cell. Why? Because it is the only cell organelle which is going to produce energy. It is going to produce energy for the complete cell process. Okay, so this energy only will be used by the complete cell. Okay, so this mitochondria, okay, it may be present. See, this mitochondria, since it is going to produce energy, okay, so it depends. Okay, the number of mitochondria different. Okay, differs based on an organism. Okay, so there will be some microorganisms contains only single mitochondria. Okay, some organisms contain only single mitochondria, like you can say green algae, unicellular blue-green algae. Have you all heard about this? BGA. Okay, unicellular blue-green algae have only one mitochondria, whereas a normal cell, whereas a normal animal cell. Okay, we are talking about animal cell. When we talk about an animal cell, they have around 50 to 50,000. Okay, they will be having around 50 to 50,000 mitochondria in a cell. Okay, per cell. Okay, per cell they will have around 50 to 50,000 mitochondria, because a cell, animal cell, is eukaryotic. Am I right? So they will be having lot of functions to do. So for that they require lot of energy. So for that so many numbers of mitochondria will also be present in a cell. Whereas blue-green algae is a prokaryote where the functions are limited, so they will be having one single mitochondria. Okay, fine. So this mitochondria are collectively called as, all these 50 to 50,000 mitochondria are collectively called as chondriome. Remember this, all 50 to 50,000 mitochondria are collectively called as chondriome. Okay, these terms are important. See guys, very important to know. NTSE and any type of Olympiads and any type of competitive exams, it will be in a MCQ type. Okay, it will be in a MCQ, that is multiple choice questions will be there. Okay, so by the time you should be very clear about the concept. Okay, so nobody is going to ask to write theory about mitochondria. Okay, so by the time you have to remember every concept, terminology is important. Okay, so for your exam point of view, for your board exam point of view, or for your school exams, you can just take these notes. Okay, you can just take these notes and you can write it whatever I'm saying. Okay, so it will be like 3-in-1 process. Okay, this videos, this NTSE series will be 3-in-1 process for you. So it will be helpful for you to attend Olympiads, and it will be helpful for attending NTSE as well as for your school exams. Okay, fine. So who first identified and termed this mitochondria? C. Benda. There is a scientist name called as C. Benda in 1897. In 1897, C. Banda termed mitochondria. Mito means thread, chondria means granules. Okay, so he founded some thread-like granules. Okay, he found some thread-like granules. Okay, so and he named that granules as mitochondria. Okay, fine. Then when we talk about ultrastructure, when we talk about the structure of mitochondria, mitochondrions are rod-shaped cells. Okay, they are all rod-shaped. Okay, so you can say rod means some vertical shaped cells are called as rod-shaped cells. Okay, and they will be bounded with double membrane. Okay, so they have two membranes. Okay, let's say like here you can see two membranes are there. One is inner membrane and another one is outer membrane. Okay, so they are all bounded with two membrane. Okay, so inner membrane, outer membrane will be smooth. Okay, and inner membrane will be surrounded with a matrix, right? Okay, outer membrane is smooth because there is no such ribosome, nothing. Okay, here see, you can see ribosomes are present, which are all freely moving ribosomes. Okay, so these ribosomes are all moving inside the matrix. Okay, so this outer membrane will be smooth, whereas inner membrane will be filled with matrix. Okay, and inner membrane will, you can see some folding. Okay, this folding, you can see this foldings, right? Okay, so these foldings are called as cristae. These foldings are called as cristae. So what is the role of this cristae? This cristae will increase the space. Okay, it will increase the space for energy production. Okay, it will increase the space for energy production. Okay, so for energy production there should be a space, right? Okay, that space will be increased with the help of this this cristae, this foldings. Okay, there will be some tubular foldings which is present inside the mitochondria, and those tubular foldings are called as microvilli. Okay, tubular folding means tube-like structure. Okay, tube-like foldings will be there, which is called as microvilli. Okay, and this villi is also going to help in the same process. Okay, increasing the space. Cristae and microvilli both are going to do the same concept. Okay, but just you can say based on shape we are differing. Okay, if it is tubular form, it is going to called as microvilli. If it is only in an inner membrane fold, then it is called as cristae. Am I right? Okay. And this mitochondria, usually it contains a transport system. Okay, it helps in electron transport system. Okay, so electron transport system means electrons will be transferred within the mitochondria. Electrons will be transferred within the mitochondria. Make it very simple. Okay, so let's say like a sun. Okay, from sunlight, sunlight will be received by a plant. Am I right? Okay, and this plant is going to produce energy. Am I right? This plant is going to produce energy. So who is exactly producing energy? Our mitochondria. So mitochondria, what it will do, it will release, it will receive all the electrons. It will receive all the electrons whichever produced by the leaf. Okay, and those electrons will be converted into energy. Okay, and that is called as electron transport system. So this is also one important point of mitochondria. So now till now you all know what is mitochondria. Mitochondria is a powerhouse of cell. Apart from that, it also helps in cellular respiration. Remember this point. Okay, it also helps in cellular respiration. Okay, it also helps in cellular respiration or electron transport system. Okay, for that electron transport system, they have one particle which is called as F0 F1 particle. Okay, they will be having some particles which is called as F0 F1 particles. You can see in this image. So this one. Okay, so which will be present near the cell wall. Okay, which is present near the cell wall. Okay, this F0 F1 particles only will release the energy. Okay, that only will release the energy. Okay, and those F0 F1 particles are also called as oxysomes. They are also called as oxysomes. Okay, so here in mitochondria, Krebs cycle will take place. So I don't want to go what is Krebs cycle. Okay, so this Krebs cycle is nothing but it is going to produce energy. Okay, so it is going to produce energy, and that energy will be finally converted with the help of F0 F1 particles, and later it will be released. Okay, so this is all going to happen inside mitochondria. This is all going to happen inside mitochondria. So mitochondria is called as power plant, powerhouse, cellular furnaces. Fine. So this is all what you have to study about mitochondria. Okay. Fine. Okay, so it is going to produce energy in a form of ATP. It is going to produce energy in a form of ATP. What is ATP? Adenosine triphosphate. What is ATP? Adenosine triphosphate. Okay, and this will be done with, this will be the process of production of, the process of producing ATP is called as oxidative phosphorylation. Oxidative phosphorylation. So what is this oxidative phosphorylation? It is nothing but production of energy. Okay, what is oxidative phosphorylation? Process of ATP synthesis. Okay, what is oxidative phosphorylation? Process of ATP synthesis. Okay, or you can also say energy production. Process of energy production is also called as oxidative phosphorylation. So this oxidative phosphorylation occurs in mitochondria. So this is all what you have to study. Okay, so I'll just give a small recap of mitochondria. Make it very simple. Okay, just remember all these things. Mitochondria, as we all know now. Okay, so after the studying, we have to remember very properly. Okay, so mitochondria, this is also called as powerhouse of cell. Am I right? And apart from that, what it is going to do, it also going to help in cellular respiration. Okay, it also going to help in cellular respiration, also in electron transport chain. Am I right? Electron transport chain or electron transport system. Okay, and Krebs cycle will take place inside mitochondria. Okay, this mitochondria numbers are different. Okay, in BGA, what is BGA? Blue-green algae. In blue-green algae, the number of mitochondria will be 1. Whereas in animal cell and plant cell, it may be around 50 to 50,000. Okay, 50,000 mitochondria will be there. Those 50,000 mitochondria are collectively called as chondriome. Okay, and it was termed by C. Benda. Okay, and the structure is rod shape, which is double membrane. Okay, outer membrane is smooth, inner membrane will be covered with a matrix. Okay, and inner membrane has a folding which is called as cristae, and there is some tubular folding which is called as microvilli. Both these foldings are going to help in increase space. Okay, it is going to increase the space for energy production. Okay, it is going to increase the space for energy production. There is one particle which is called as F0, F1 particle, or also called as oxysomes. So this particles helps in releasing energy. Okay, this helps in collecting all the electron particles, and it will finally release as a energy. Okay, so here only you can say in F0, F1 particle, in F0, F1 particle only oxidative phosphorylation takes place. Oxidative phosphorylation. What is oxidative phosphorylation? Oxidative phosphorylation is the process of ATP synthesis. Okay, ATP synthesis, that is called as oxidative phosphorylation. Okay, and inside Krebs cycle will take place, and the functions they are all called as power plant, powerhouse, cellular furnaces. Okay, and oxidative phosphorylation takes place in this. Okay, so the person who founded the mitochondria will also undergo cellular respiration. The person who founded mitochondria will also undergo cellular respiration, and that guy name is, the scientist name is Hogboom. Okay, the scientist name is Hogboom. Okay, so two scientists we have to remember for mitochondria. One is C. Benda, who termed, okay, who first observed and termed. Okay, and the function mitochondria involves in cellular respiration, that was discovered by Hogboom. Okay, Hogboom, Hogboom, whatever you say. Okay, that is what about mitochondria. Fine. So with this, we are completing mitochondria. Last cell organelle of this chapter, we'll be completing with plastid today. Okay, and in the next video, we'll go with Golgi complex, one of the complicated cell organelle. Okay, we'll go with a separate video for Golgi complex. Okay, now we'll complete with plastid. What is plastid? So this plastid is one of the unique cell organelle. Unique means different. Okay, you can say it is different. Unique cell organelle which is present only in plant cells. Plastids are the cell organelle which will be present only in plant cell. Okay, and they will be present in all plants, and they are all double membered. Okay, and they are all double membered. So till now, how many double membered cell organelles we have found? Nucleus, plastids, mitochondria. all this nucleus, plastids and mitochondria are all double membered. Okay, they will be having outer membrane, inner membrane. Okay, so who first termed the plastid? Ernst Haeckel. His name is Ernst Haeckel. Okay, and he did this in 1865. Okay, he gave the term plastid, and this plastid is also one of the largest cell organelle. Remember this, largest cell organelle in a cell is plastid. Okay, smallest cell organelle in a cell is ribosomes. Remember that. Fine. Okay, it is also called as largest cell organelle. So these plastids are usually self-replicating. Self-replicating means it will undergo self-division. Okay, this plastids will undergo self-division. Okay, so one plastid will divide into two plastid, two will divide into four, like that. Okay, so that division will take place. Okay, and that is how same like mitochondria will also do. Okay, mitochondria will also undergo direct cell division. I forgot one important thing. This mitochondria is a cell organelle which contains its own DNA. Remember, this is very important. I'm sorry, I forgot it. Okay, so the mitochondria is a cell organelle which contains its own DNA. So that means the normal DNA which is present inside nucleus cannot control. It cannot control the mitochondria. The mitochondria will be controlled with its own DNA. Am I right? So here plastids all like going to be under the control of nucleus. No worry. Okay, so but they will be self-replicating like a mitochondria. Okay, fine. So there was a scientist, his name is Skimper. Okay, there was a scientist name is Skimper who divided this plastid, who gave the types of plastid. Okay, he founded three types of plastids. One is chromoplast, another one is chloroplast, and another one is leucoplast. So this chromo means color. Okay, chrome means color. Okay, chromoplast means which have all colors except green. Okay, which have all colors except green. Okay, whereas chloroplast which contains green color plastid, which is also called as green color plastid. Okay, and leucoplast means colorless. Leucoplast means colorless plastid. Am I right? Okay, so this leucoplast you can find in roots. Okay, this leucoplast you can find in roots, and this chloroplast you can find throughout the throughout the plant. Okay, you can find throughout the plant. And chromoplast you can see this in leaves as well as flowers. Okay, you can see this in leaves and flowers. Okay, you can see yellow color flower, pink color flower, white color flower. That is all because of chromoplast. Okay, and the green color is because of chloroplast. Okay, and the green color leaves, green color stem is all because of chloroplast. And leucoplast will be present, which will be present in roots. Okay, which is colorless. Then you can ask me, sir, then brown color is the roots are brown in color. No, that brown in color is because of soil. Okay, they are all dissolved. The roots are dissolved in soil, so they are brown in color. Okay, or else like roots doesn't have any color. Okay, so plastids are also double membrane, but they don't have any cristae. So what is the plastid function of plastid? They helps in entrapping solar energy. Okay, so they will entrap sun energy. Okay, they will collect the sunlight. Okay, and they will utilize, they will produce food. Okay, they will produce food for the plants. Okay, and this chromoplast will involve in various color formation. Okay, and it also, this color formation is also help in attracting pollinators. Okay, it also helps in attracting pollinators. Who is pollinator? Let's say honeybee is a pollinator. Okay, honeybee is a pollinator. What it will do? It will take a juice from plastid, from a male flower, and it will take it to the female flower. Okay, so these honeybees are called as pollinators. Okay, which is going to be a mediator between male flower and female flower. Okay, so for attracting insects, okay, colors are very important, and that process will be takes place by chromoplast. Okay, so this is all about plastid. So in plastid, what we have to remember? Let me first erase all these things and we'll have a short recap. So plastids is a unique cell organelle which is present only in plant cell. They are double membrane. It was identified by Ernst Haeckel, and this is the largest cell organelle which will replicate, which will divide by itself. Okay, and it is divided into 3 types by Skimper. Okay, so what he gave a colored plastid. He identified colored plastid, then he named it as chromoplast, and he also found a green color plastid, and he named it as chloroplast, and he found a colorless plastid, okay, and he termed it for leucoplast. Okay, so this chromoplast will be present in flowers, okay, and this chloroplast present in leaf and the whole body of the plant. Okay, and leucoplast will present in roots. Okay, will present in roots. So this colored chloroplast will also helps in attracting pollinators. Okay, so this chloroplast, what it will do? It will entrap sunlight. That means it will receive sunlight, and it will help in producing food. Okay, it helps in producing food. So this is the structure of chloroplast. Okay, we'll study these structures in detail. Okay, whenever we'll be having a separate segment for diagram based study, conceptual studies. Okay, by the time we'll have all this diagram based concepts. Fine. So this is all about plastids. Okay, I hope you all are clear with the concepts now. Okay, so today we have studied about core cell organelles. What are all that? Nucleus, endoplasmic reticulum, mitochondria, plastid, okay, and ribosomes. Am I right? So 5 organelles we have studied. So just go through this video once again, just pass and recollect all the information whichever we studied, take a note of it, okay, and it will help you in a lot of way for your exams and as well as for your competitive part. Okay, so with this I'm winding up today's session. I hope you all enjoyed this session, and next lecture will be about Golgi complex and rest about the plant. Okay, cell. Okay, so we'll be completing cell maybe in the next few lectures. Okay, so we'll complete very deeply. Okay, I will teach you whichever is required for clearing NTSE. Okay, and also is going to help you for board examinations as well. Okay, so our motto is think big, dream big. So don't forget to like and share and subscribe this channel. Okay, so if you are doing all these things, it will be motivating me a lot. Okay, so I can also move further for the betterment. Okay, so with this I'm winding up today's session. I hope you all enjoyed this. Okay, so we'll meet you in the next video. Bye.